Hello everyone, myself A. Sudha from 1st MSc Applied Microbiology, Secretariat College, Tirupattur. My topic is about gram-negative bacteria, Vibrio cholerae. Let's see the general characteristics of Vibrio cholerae. Gram's classification, it shows gram-negative, the shape is comma-shaped curved rods, bacilli, the motility is motile, capsule and endospores are absent in Vibrio cholerae. Respiration is through aerobic or facultative anaerobic. Optimum temperature is 37 degrees Celsius. It can also able to grow between 20 degrees Celsius to 45 degrees Celsius. The optimum pH is 7.6 to 8.6. Habitat brackish water, that is high salinity water and salt water. Vibrio cholerae was discovered by Robert Koch in 1884. Classification of Vibrio cholerae. Vibrio cholerae is classified into two types, Vibrio cholerae O1 and Vibrio cholerae O139. Vibrio cholerae O1 is subdivided into serotypes and biotypes. Serotypes as Inba, Agua and Hikojima. Biotypes as classical and E1 tar. The pathogenicity of Vibrio cholerae. Disease transmission. Vibrio cholerae infection are mainly transmitted by ingestion of contaminated food and water. Incubation period, that is the time taken to exhibit its symptoms, is about 2 to 3 days. Virulence factors of Vibrio cholerae, that includes flagella which facilitate motility, cholera toxin, helps in hypersecretion of electrolytes and water, toxin co-regulated pilus that helps in adherence to the intestinal mucosal cells, chemotaxis protein that is adhesion factor, accessory cholera enterotoxin increases intestinal fluid secretion, zonula occlusions toxin increases intestinal permeability, neuraminidase modifies cell surface to increase GM1 binding sites for cholera toxin. Pathogenesis of Vibrio cholerae. Vibrio cholerae are able to adhere to the mucosal cell layer by means of the TCP encoded by the TCP genes, that is toxin co-regulated pilus, and chemotaxis protein encoded by the CEP genes. Thus the TCP is important both as receptor for cholera toxin. Cholera toxin binds to the ganglioside GM1 receptors on the intestinal epithelial cells. The active portion of the cholera toxin, that is A subunit, is internalized, interacts with G protein that control adenylate cyclase, leading to the catabolic conversion of adenosine triphosphate to cyclic adenosine monophosphate, that is CAMP. This results in a hypersecretion of water and electrolytes. Severely infected patient can lose as much as 1 liter of fluid per hour during the height of the disease. Such a tremendous loss of fluid would normally flush the organism out of the gastrointestinal tract. The non-adherent strains are unable to establish infection. In the absence of cholera toxin, Vibrio cholerae O1 can still produce significant diarrhea through the action of the zonula occlusions toxin and accessory cholera enterotoxin. As the name implies, the zonula occlusions toxin loosens the tight junctions of the small intestinal mucosa, leading to increased intestinal permeability, and the enterotoxin produces increased fluid secretion. Clinical diseases caused by Vibrio cholerae include cholera and gastroenteritis. The clinical manifestation of cholera begin an average of 2 to 3 days after injection of bacteria with an abrupt onset of watery diarrhea, vomiting and nausea. As more fluid is lost, the stool specimen becomes colorless, odorless, free of proteins and speckled with mucus that known as rice water stools. The resulting severe fluid and electrolytes can lead to dehydration, painful muscle cramps, metabolic acidosis, hypokalemia and hypovolemic shock, cardiac arrhythmia, renal failure. Then gastroenteritis. Vibrio cholerae causes gastroenteritis. Milder form of diarrheal disease can occur in toxic negative strains of Vibrio cholerae. The toxic negative strains of Vibrio cholerae can also cause extraintestinal infections such as septicemia, particularly in patients with liver disease. Then laboratory diagnosis of Vibrio cholerae. It includes microscopic examination. Gram staining, it shows pink color gram negative gamma shaped curved rods. Motility test, it shows motile. Colony morphology on culture medium. In nutrient agar, colonies are moist, translucent, brown disk with a bluish tinge in transmitted light and a distinctive odor. In McConaughey agar, it shows lactose fermenting pink colonies. The selective medium for Vibrio cholerae is thiosulfate citrate bile salt sucrose, that is TCBS agar. It shows yellow colored colonies. Biochemical test for Vibrio cholerae. Catalase test, it shows positive, oxidase test positive, urease test negative, indole test positive, methyl red, that is MR test positive, Vogus proscarer VP test negative, citrate utilization test variable, cholera red test positive, ONTG disc test positive, syringe test positive. Treatment and preventive measures for Vibrio cholerae include fluid and electrolyte replacement are crucial. Antibiotic example azithromycin reduces the bacterial burden and exotoxin production as well as duration of diarrhea. Improved hygiene is critical for control. Combination of inactivated whole cell and cholera toxin B subunit vaccine provided limited protection and herd immunity. Thank you for listening.